Hey guys, so in this video, we're going to start going over information that is important for that second lab practical. In this video, I'm going to be answering basically every single question that we have on that study guide that was given to us. So be sure to write this down. The PowerPoint that you see here, I did make a few changes to it, but the original one can be found on the Biology Resources website, as well as I did upload it to our Canvas course. So we're going to be starting with Lab 6, which is going to have a focus on enzyme activity. So the first thing that I want to do is actually define what an enzyme is. So if you guys remember, enzymes, they are proteins that are biological catalysts, and what that means is that they're going to help to regulate the rate of reactions, and they do this by lowering activation energy. When we have a lowered activation energy, our reactions, they can occur faster and more efficiently. In this lab, we experimented with the enzyme catalase. Catalase is an enzyme that is found in almost all living organisms except for obligate anaerobes. An obligate anaerobe, it is an organism that can only grow in the complete absence of oxygen. So catalase actually works by decomposing hydrogen peroxide into water and oxygen. The source of catalase in this experiment was sheep's blood. Let me write down here. Source of catalase. It was sheep's blood. So a substrate, it is the substance on which an enzyme is going to act on. So in this case, when we're looking over here at this figure, the enzyme that we're talking about is going to be catalase, and the substrate that we're talking about is hydrogen peroxide. Hydrogen peroxide, it is our substrate, and hydrogen peroxide, it is actually toxic to our cells. So catalase is going to convert hydrogen peroxide into water and oxygen, which are two completely harmless byproducts. So when we are analyzing a chemical reaction, no matter what chemical reaction it is, the substances that we have on the left side, they are always going to be our reactants. And the substances on the right side, they're always going to be our products. So if you guys are asked what the reactants and products are, now you know. So the way that we measured catalase activity, it was actually by observing the amount of oxygen gas that was produced during the reaction. And the reason that we do it that way is because the rate of oxygen production, it is directly proportional to the enzyme's activity when we are talking about catalase specifically. We used a biology gas pressure sensor. We connected it to a computer and were able to measure oxygen gas production that way. You guys may have noticed bubbling in the test tubes. The bubbling was actually visible evidence of oxygen being released during the reaction. So we had a few different independent variables in this lab, and those independent variables included pH, temperature, enzyme concentration, substrate concentration, and the presence of inhibitors. These are considered our independent variables because we manipulated them to observe how they affect catalase activity. So before we actually start to talk about this pH graph, I want to quickly explain how the experiment was set up. So to test each condition, whether it was pH, temperature, enzyme concentration, or anything else, we had to use separate test tubes, and we prepared them specifically based on what the variable was that we were testing. So for example, when we were testing pH, we used three different test tubes. we labeled them with a different pH level. So let's say this one had pH 4, this one had pH 8, and this one had pH 12. In each test tube, we added 2 milliliters of the corresponding buffer solution. Then we took a clean pipette, we added 2 milliliters of sheep's blood. And then finally, we took 2 milliliters of hydrogen peroxide and added them to the test tubes. Remember, sheep's blood was the source of catalase, and hydrogen peroxide is our substrate. Once we had all of those components that were added, we began to measure the production of oxygen gas. And we did this by using a gas pressure sensor. So basically, it was like a little cap that we put here. And this cap, it was connected to a computer. And this computer, it started to read different levels of activity. So this was the same setup process that was repeated across the different variables like temperature and concentration, but all we did was adjust the conditions as needed. So for example, we followed a similar procedure for all of these independent variables. So adjusting that setup, it was dependent on what we were testing. When we were testing temperature, we placed the test tubes in water baths to set different temperatures. So when we tested for substrate or enzyme concentrations, we varied that amount of hydrogen peroxide or sheep's blood accordingly. So now we can start to talk a little bit about the graphs. The graphs, they help us visualize the effect of each variable. But I need you guys to remember that our x-axis, right? So our x-axis, it is always going to show our independent variable. Remember that our independent variable is what we are changing. And our y-axis, it is always going to show the dependent variable. The dependent variable is always what we are measuring. And in this case, what we're measuring is always going to be oxygen gas production. So this first graph is showing us the effect of pH on catalase activity. So when we are looking at pH 4, we see no catalase activity. And that is because the enzyme, it is denatured because the conditions are too acidic. Remember, so at pH 4, it is too acidic. And what that means is that we have too much H plus, too much protons, too many hydrogen ions. When we move to pH 7, we actually see that we have the highest activity, and that gives us an activity of 2.212 kPa. That actually makes this the optimal pH. When we look at pH 10, we see that we still have activity, but it was reduced. And by the time that we get to pH 12, again, we have no activity. But this time we have no activity because it is too basic. What that means is that we have too many OH, which are also going to denature the enzyme. So here we have denature. We have denature at pH 12, and then at pH 10 we have reduced activity, and at pH 7 we have optimal activity. So when we're looking at the effect of temperature on catalase activity, when we are at 0 degrees Celsius, we have really low enzyme activity. Our molecular motion is slow. 0 degrees Celsius is considered ice. Well, not is considered, it is ice. So we just have slow movement. When we get to about 21 degrees Celsius, we get room temperature. And we do see that our activity did start to increase. But when we reach 40 degrees Celsius, what we have is the peak of catalase activity. And when we get to to temperature 60, our activity, it drops off sharply because we have heat-induced denaturation. So we actually know that 37 degrees Celsius is its optimal temperature, but we didn't specifically measure 37 degrees Celsius. Based on the curve, we can infer that it's just below 40. So the effect of substrate concentration on catalase activity. So when we're looking at 0, we have no enzymatic activity, and that is because enzymes need substrates in order to function. As we have this increase in substrate concentration, our enzymatic activity does increase and then it plateaus. The plateau happens because the enzyme becomes too saturated. That means that they are all occupied and working at full capacity. So the effect of enzyme concentration on catalase activity. As we start to increase enzyme concentration, going this way, our activity starts to increase again, and then it plateaus. By the time that, at its plateau, what it does is it suggests that all the available substrate has been used. And at that time, we actually have no decline in activity because that means that we didn't add any more substrate beyond the enzyme's capacity. And then we have the addition of copper sulfate. Copper sulfate is the inhibitor that was used in this experiment. I think it would be wise that you guys remember that the formula for copper sulfate is CuSO4. So, our experimental control, it was set up without any enzyme or substrate added to it. And that is what gave us the baseline that allows us to compare how each condition affected the reaction. So when we added our copper sulfate, our enzymatic activity, it decreased significantly. And that is because that inhibitor, it either blocked that active site or it altered the enzyme structure, making it unable to bind to the substrate effectively. Remember that an enzyme inhibitor, it can work through competitive or non-competitive manners. So that's actually all for Lab 6. I suggest that you guys go in and look at your lab notes and everything and follow along. But I answered all the questions that were in the study guide, so at the very least, please study that. In the next video, we're going to be going over Lab 7, which is all about photosynthesis and respiration.